Thank you very much, and I'd like to tell everybody I appreciate the opportunity for Lumithera to present. Today we're going to talk a little bit about our LightSight 3 U.S. pivotal trial, but I also want to have people alerted to the fact that what we're not going to talk about today is we've acquired 2 companies in the last 6 months. One to allow us to monitor our treatment or our therapies, and another one to identify patients very early on with dark adaptation wearable. But the focus for today's study is on our pivotal trial data, and this is the Valeda light delivery system. It's a multiple wavelength approach to stimulate retinal cells to improve blood flow and energy production at the cellular level. It's targeting the mitochondrial enzyme cytochrome C oxidase, and that's key in regulatory pathways of metabolism. The device is approved in Europe as well as in Latin America. We've got about 7,000 patients that we've now tested commercially in Europe and in South America. The device provides a non-invasive treatment for each eye, individually or both eyes. It's a little over 4 minutes. We go through a series of about 9 treatments over a very flexible 3 to 4 weeks, and that stimulates the pathway and allows us to have long-term benefits. It is a therapy, and so we have to do repeat treatment. We do that about 2 to 3 sessions a year, every 4 to 6 months. The U.S. trial was enrolled 100 subjects, 148 eyes qualified for the inclusion criteria. The other 52 eyes or non-study eyes were followed, and all the endpoints were measured on those eyes as well. You can see here the retinal centers were top retinal centers, including Stanford with Diana Doe and Nora Ladd at Duke. And the imaging center that managed all the treatments was Glenn Jaffe at Duke Imaging Center. The study design was a double-masked, randomized, sham-controlled, parallel group multi-center trial to look at the safety and efficacy of photobiomodulation with patients with intermediate dry age-related macular degeneration. So unlike some of the trials that we've heard today, this one is really looking at earlier patients with intermediate. They had to have at least one single large drusen. Patients were excluded if they had central fovea GA, and the patients had to have initial vision loss 2032 to 2100. So these are patients that are pretty good in terms of vision. They're starting to show signs of disease and are still pretty active. We randomized them 2 to 1 into the active versus a sham, and to do a masked study in light is not easy. So we cannot, reduce the light to 0. That would be the perfect placebo, but the patient would know. So what we do is reduce the wavelengths that are visible by about 50 to 100 fold, so it is an active control. And those patients were the sham treatment group. We went through 6 rounds of treatment. I'm happy to say today all the patients have finished their 6th round of treatment. We're approaching the 24-month data. We've already got the 13-month data that we've presented at some of the meetings this year, and the 24-month data, we've got about 12 patients left to exit the study for us to close in the end of January. So we're very excited about the future for the study. The study was basically 100 patients, 68% were female, mainly Caucasian. The mean age was around 75 years of age, and about 5 years from diagnosis of AMD. In the United States, we did stratify all the sites by AREDS, whether or not they were on AREDS. They were supposed to maintain their AREDS vitamins, and about 86% of the patients were on AREDS supplements. These patients, as you can see, started out with pretty good vision. So they're at a 70-letter baseline, which means that about 70% of our patients had 20/40 or better vision to start with. What you see in the green is a sustained improvement in BCVA compared to the sham in gray. The pre-specified endpoint was month 13, and we saw statistically significant improvement over the sham treatment with a p-value of 0.02. Importantly as well, from the baseline eyes at 13 months, we got a sustained improvement in BCVA with a mean of over 5.5 letters, and that was significant, a p-value of 0.0001. To look at that a little bit closer, we have here also the non-study control eyes. Now, when we look at this, the non-study control eyes by definition are either those that are too early or too late, or maybe have already converted to wet. So when we had 52 patients, 16 of those were wet eyes. We removed that, and then we looked at those patients that were actually good vision. 75 letters or more. There were 16 patients, 3 which converted and 1 which had a glaucoma, cataract that extended some vision loss. So when we look at a pure 12 patients that had a good eye, that were in the non-study, that were too good to look at, we actually saw what you see in the literature, about a 2 to 3 letter loss in those patients. So it's not just about letter gain, it's also slowing down the letter loss in these patients. And that was shown here. If we look at the distribution of the BCVA improvement or loss, we saw about twice as many patients that had letter loss in the sham versus the active. And you can see the shift to the right in terms of BCVA letter improvement, with many patients getting 5 to up to 20 letter improvement. If we look at those 4 groups, we had about 55% of the PBM patients that had at least a 5-letter gain with a mean of 9.7 letters, almost 2 lines, and 26% that had a mean of 12.8 letters. So considering these patients start with a mean of about 70 letters, this is a very nice improvement in BCVA. What we've seen in our 3 clinical trials also is that we're stabilizing drusen deposition. In some of the patients with later stage, we actually see reduction in drusen deposition and improvement in contrast sensitivity and BCVA. In this case, we are now intermediate, so we have smaller amounts of drusen, but you can see over the course of the 13 months in the sham, we continue to see increases in drusen, where we basically have seen no change in the underlying amount of drusen now in central drusen volume. And at 13 months, you can see the difference between the two. We also looked at the safety as we were putting this together for the FDA. We looked at the patients that had no GA to begin with and how many cases at 13 months now showed some signs of new onset GA. And what we saw was about 10% of the sham patients had new GA, or only 1% of the PBM. So we went back and looked at other 2 trials, and it seems to be holding. We've got about 5-fold more patients in the sham that moved to new GA in the time period of 10 to 13 months. So what I'd like to say in summary is we've got a very clean product here in terms of we've seen no safety profile issues. In fact, we have never had our independent safety data board have to call a meeting over the 24 months. We've got a very nice improvement in BCVA across the board. We've seen some reductions in the letter loss, as well as reductions in the amount of GA that forms in naive GA patients over the 13 months, and clearly the similar effects that we're seeing on drusen is stabilizing or reducing deposition. So lastly, I want to talk about very quickly the reimbursement of this. This is a product that can provide a very nice quality, and what we've done is modeled, looking at the literature out there, a 75-year-old person and the vision loss over the course of their life. And what's important about that is if you look at these clinical trials, on the right is basic standard care, which shows dry AMD in green, the amount that convert to GA or in gray, in yellow is the amount that convert to wet, and in the dark green is when they become legally blind. If you look at a 5.5 letter gain early in these patients, you can see the benefit you can have. And so I just like to summary that it's very important that we look at, if we can treat these chronic diseases early, the big impact we can have on the quality of life for the patients. And so with that, I'd like to say thank you very much.